Thank you. I'm honored to be here today, and thank you, Jeff, for inviting me. So today we've heard a lot about engineering unit processes, but we haven't heard too much about natural systems and natural unit processes for treating water. And in Renew It, we work on both engineered and natural unit processes. And today I'm going to tell you a little bit about some of the work we're doing on stormwater in Renew It. Before I begin, I would like to acknowledge Sanjay Mohanty, who is the postdoc who did all of the research that I'll be talking about today. So we've heard this morning about stormwater from two different speakers. We heard David Sedlak mention it, and we heard Jim McDaniels speak about it in LA. And urban stormwater is basically a lost water resource. Under natural conditions, it might infiltrate into the groundwater, where it can represent a drinking water supply. And because of impervious surface coverage, a lot of the water is directed out of urban areas and discharged into coastal waters in coastal cities or into other surface receiving waters. At the same time, though, stormwater is a major source of surface water contamination. It contains a lot of different contaminants, which represents a challenge to using it as a water resource. So let's consider a case study in Monterey, California, located close to Stanford. Here they get a lot of their drinking water from the Carmel River and from extensive groundwater pumping adjacent to the river. And recently the Carmel River has actually gone dry, and it can be dry 5 to 6 months of year, which is obviously a huge burden on the ecosystem. And so they'd like to restore the river back to what it used to be and have surface flow in the river throughout the year. And so the State Water Board has actually issued a cease and desist order for the County of Monterey, and they're basically in this very serious situation where I think it's around 20 to 30% of their drinking water supply will no longer be available to them. So they're thinking about regional solutions, which include water reuse, desalination, but they're also seriously considering aquifer storage of stormwater, because there's a lot of stormwater in this area that just goes directly out into the ocean. Now, at the same time, stormwater is highly polluted. According to the EPA, it's responsible for 10% of all of the 303D listings from the Clean Water Act of impaired water bodies. And of those impairments, pathogens and indicator bacteria are the top cause of impairment. About 30% of impaired waters are impaired with respect to these contaminants. In urban stormwater, we can find E. coli and enterococci, the two regulated fecal indicator bacteria, at concentrations between 1 and 1 million per 100 milliliters, which is the highest value is similar to what you would see in raw sewage. So it's important to control stormwater contaminants for reasons related to surface water quality, like TMDLs, as well as MS4 permit compliance, but also for considering infiltrating this into the ground for potable and non-potable uses. Then we need to be aware of the pollution in stormwater. So there are several types of low-impact development BMPs that are available for treating and capturing stormwater. And these are basically living systems. So the four listed here are common ones. The bioswale is basically a vegetated ditch. And then the bioretention, media filter, and biofilter are all related to the diagram here. So as shown here, this is a biofilter, and in some places it can be called a rain garden. And basically you have a ponding zone with some vegetation, some mulch where the vegetation is growing, and then some speck soil, which might be a combination of sand and compost, underlaid by some gravel. In this particular diagram, this water that percolates through here can then overflow back into the storm sewer, so it's not infiltrating in this system. It's just hopefully slowing down the stormwater flow, as well as removing some contaminants during the filtration process. If it were a bioretention system, then we would have infiltration, and if it were a media filter, it wouldn't have any vegetation. So these things are living systems. They are very complex, heterogeneous. They might have animals living in them, lots of plants, of course, microorganisms. So actually using these for an engineering unit process can be very challenging. If we go to this really great database, it's the International BMP database put together by Enhnc and other consulting agencies. They've compiled data from all around the world on the performance of these BMPs in the field. And actually, there are only 3 sets of data available that have evaluated the performance of a biofilter in the field for removal of E. coli bacteria. And so this plot shows a box and whisker plot of what's going into the biofilter versus what's coming out for the 3 different ones, 1, 2, and 3. And what you'll see here is that there's one biofilter where what comes out, the concentration of E. coli, is much lower than what came in, significantly lower. In one of them, there was no significant difference. And in one, it was significantly higher coming out than going in. So not achieving what is hopefully the design criteria, which is to remove these sorts of organisms from stormwater. This is a great resource, this database. I've also looked at some data on nutrients, and unfortunately, things like biofilters are also not that successful at removing nutrients, and they have similar results as those shown here for E. coli. Despite this, stormwater BMP installation is occurring in major cities, and there's basically the community can really get around the implementation of these systems, as we heard earlier this morning in the presentation in LA. And the idea is that in people's neighborhoods, they can get really excited about going from something that looks like this, where stormwater is waste and it's being directed into a surface water where it's potentially contaminating that surface water and putting it on a 303D list. They want to move from something like this to this, which is a really nice looking rain garden where the stormwater can infiltrate. And ideally, as engineers, we'd like to think that this could be engineered to remove contaminants and actually infiltrate into the groundwater where it can be available as a potable or non-potable resource. So communities can really get behind this greening of the stormwater infrastructure, and it can also be potentially useful for TMDL control, MS4 permitting, capturing the first flush, all with the goal of reducing contaminants as well as augmenting groundwater. And of course, the urban aesthetics is very important as well. So that's great, but the truth is that these are living systems. They're living unit processes, natural systems. And so based on the data that's available, the performance of them is quite inconsistent. And so given that there is future planned widespread installation of these systems, we really need research to help us improve their design and make their performance more predictable. So the idea is that if you have a system like this and you know the concentration coming in, then you can predict what the concentration coming out will be. So in Renew It, we've been working on these systems and trying to understand how we can engineer them to make their performance more predictable. Within the center, we've been working on both chemical and pathogen removal, and today I'm going to talk about some of the experiments on pathogen removal. And our approach is basically to start with lab screening of different types of geomedia that might be able to improve the removal of target contaminants from stormwater. So in lab screening, we're working with simulated stormwater, adding contaminants, and seeing how well they perform. The next step will be to move on to preliminary field testing using authentic stormwater and incorporating some more complexities in the system, such as vegetation and microorganisms, and then moving to the test bed to actually look at performance in the field. So I'm going to tell you a little bit about some of the research that's gone on in the center regarding removal of E. coli with different geomedias. So the 2 research questions that guided this work were, can addition of geomedia to biofilters improve their performance? And how does intermittent flow affect performance of biofilters? We know that rainfall doesn't continuously fall. In fact, in California, it seems like it doesn't fall anymore. But there are actually pauses between rain events, and the drying and re-wetting of these systems might be able to mobilize bacteria. And so we looked at that as well. So the first question, can addition of geomedia to biofilters improve their performance? So we looked at different geomedia, and I have a list in the next slide, but the geomedia we use basically consists of biochar, which takes a product like wood and it undergoes pyrolysis at a high temperature, different temperatures, but under anaerobic conditions. And then iron, and in particular iron filings. So this is how we carried out the work. We did column tests like the ones shown here, which are pretty basic. And we tested different geomedia, quartz sands, iron oxide-coated sands, biochars, and iron fillings. We used a synthetic stormwater, which represented a complex mix of ions and nutrients, and it had a relatively high ionic strength. We tested it with and without NOM. We used E. coli as our model bacteria. And the operation of these columns for this first hypothesis was basically to introduce the synthetic stormwater and then introduce the stormwater with E. coli in it and look at breakthrough and see which geomedia removed the most bacteria. So of course, here's some example data. It shows C over C naught in the effluent of the column, and this is time in hours. And we injected the bacteria-laden stormwater up until 3 hours and then sterile stormwater after that. And you can see that in this particular experiment, which shows triplicate results from sand, just sand alone with E. coli and no NOM in the stormwater, that you see pretty high breakthrough of bacteria. About 50% of what came in was coming out, so not very much removal. Here's some example data with a biochar. The same conditions, and this time on this scale at least, which is the linear scale, you can't see any breakthrough occurring. So we have significantly greater removal with biochar compared to sand. And this, I'm sorry, this is 5% biochar, and the rest is sand. And then one more example data before I show you a compilation of the results. This shows the same experiment in the previous slide, except this time there's NOM in the stormwater. And we see the biochar is able to sequester fewer E. coli when NOM is present, presumably because the NOM is competing for sites on the biochar for absorption of E. coli. That's one reason of many. So this is a summary of the results from these geomedia experiments. So on this axis are the different geomedia that we've tested: sand, iron oxide-coated sands with goethite, iron oxide-coated sands with hematite, three different types of biochars, 5% by mass in the column, and then iron filings with different percentages in the column. And without NOM is shown with the white bars, and you can see that as we move down here, we're getting more and more removal, with iron filings removing the greatest amount of E. coli at our detection limit. And then when we add NOM, 20 milligrams carbon per liter of NOM, that removal is reduced. But we still have the best results when we mix our sand with the iron filings, but we also have fairly good results with the biochar as well. Okay, so the second question that we looked at for this project is the effect of the intermittent flow on the performance of biofilters. Now we know that for colloids that if you have unsteady flow conditions, that they can scour colloids that are attached to grains by 3 different mechanisms. You can have surface detachment due to chemical perturbations where the adsorbed colloids, or in this case bacteria, just come off the surface. You can have shear mobilization by changing velocities. You can have thin film expansion, releasing colloids trapped in a thin film, and you can have air-water interface scouring. And we know this happens for colloids as well as bacteria under some conditions. So to test this, we used the same columns that we used in the previous experiments for all the different geomedia, and this time we subjected the columns to intermittent flow. So after they were contaminated with E. coli in the first half of the experiment, we stopped the flow. We let the water drain for 30 minutes, as it might drain after a rain event, and then we applied 2 more pore volumes of stormwater without any E. coli, then let it drain again and repeated, and captured the effluent to look at what came out. So this is the same type of plot for E. coli with sand and no NOM, and the red arrows show when the intermittent event started, or not started, but sort of the middle of the intermittent flow event. And you can see the pulses of bacteria coming out of the column. Presumably the E. coli are being scoured by an air-water interface or being released by one of those 4 mechanisms. If we look at the biochar column, this is 5% biochar and sand. We don't see the same mobilization by the wetting front that we saw with just sand. And now if we add NOM, we see that we do see some release of the E. coli. Maybe the E. coli are more weakly attached to the biochar when the NOM is present, so they're more easily scoured by that propagating air-water interface. So this is a summary of the remobilization results. This shows the same geomedia that was in the previous plot like this. And then on here, on the bottom axis, on the x-axis, is the percent of attached E. coli that were remobilized during the 2 intermittent flow events. So what's important to note here is that there are a number of geomedia where it looks like it's 0, where it is 0, where there is no mobilization of bacteria under the intermittent flow event, in particular the iron filings. And the biochar performs very well with no NOM, but when NOM is present at that high concentration, we do see that there's some scouring of the bacteria. And of course, sand is not very efficient at all at retaining anything that it sequestered from the stormwater initially. Okay, so for these 2 research questions, we found that geomedia can enhance the retention of E. coli in this model biofiltration system, although the next step really is field testing and also comparing with standard bioretention media that consultants are using in the field. And then further testing with enterococci, which is a gram-positive organism and also highly regulated in surface water, would be useful. And then for the intermittent flow events, we found that the attached bacteria can be mobilized by the wetting front during these unsteady flow events, and that mobilization can be reduced with the use of geomedia, but NOM is a problem because it does increase mobilization, and we know NOM is present at high concentrations in stormwater. Okay, so future work, I mentioned already some of these things, incorporating standard bioretention media into the experiments. In talking with Geosyntec, they've been using things like peat moss and zeolite in their biofiltration systems because they're very good at removing metals. And so it would be interesting to see how well these perform for removing bacteria. We're working right now on studying what happens when you actually ripen these columns by having microorganisms that are present in stormwater actually start growing in them. And we're also going to be doing pilot scale research. And just to take a step back, the reason that we're doing all this again is to make this biofilter performance be something that can be consistent and predictable, and hopefully be able to do modeling so that we'll know in advance how well they'll perform under certain conditions. So let's take a step back and start and talk about where we started here, where I started this talk, and talk about Pacific Grove, California. So here's a location where their drinking water source, one of their major drinking water sources, is basically going to be cut off. They can't do any more groundwater pumping next to the Carmel River, and they won't be able to get any water from the Carmel River. At the same time, they're right adjacent to the Monterey Bay Sanctuary. It's an area of special biological significance, and it's contaminated with stormwater, and it exceeds bacterial standards all the time. And they have very strict regulations on all stormwater discharges there along this area of special biological significance. So this is a location where it seems like it would make a lot of sense to do stormwater infiltration. It can provide co-benefits in addition to providing a drinking water resource and improving stormwater quality before it's discharged into the ocean. It can provide urban aesthetics and also public education to let people know, what's going on with the water and how the water cycle works. However, ensuring good operation of these systems is really important to prevent unintended consequences like contamination of groundwater, and more research is really needed to get to that point. So again, I'd like to acknowledge Sanjay, who did the experiments that I presented, Cara Nelson and Andy Turkelson, who are collaborators on this project, and then Eric Strecker and Brandon Steitz at Geosyntec, who I've talked a lot about regarding these BMPs, and also funding from the Renew It Urban Water ERC. And I'll be happy to take any questions.
Okay, questions? Where's the microphone, people? There's one over there. Okay, so we got the microphones ready. Okay, right up here, Mark Wiesner. He's always got a good question.
Okay. I don't know if I have a good question, but I have a question. So it's kind of two parts. First of all, how are you measuring E. coli? Can you differentiate between just bacteria that are removed versus inactivation of bacteria because of the role of the metallic iron? Mm-hmm. And then given that the hematite didn't work very well and the metallic iron was working really well, and that the metallic iron is going to turn into iron oxides over time, what do you do with the system?
Yeah, so the first question, how are we measuring the E. coli? We're just using culture-based methods, so we haven't tried to figure out what's been inactivated versus adsorbed, and as you probably know and have thought about, that's very complicated to do, but that's something that we could do. And then in terms of the iron filing results, the next step obviously is to do like some aging experiments and see what happens over time, because that's a very valid point. Yeah.
All right, here we go, back here, Bill Ball.
Hi, Ali. Nice talk. I have a similar question about comparing the hematite and the iron filings, and also comparing the iron filings to other iron filings in the future. How did they compare in terms of total surface area, particle size, distribution within the media, those kinds of things? How did you control that?
So the iron filings were sieved so that they were the same size as the quartz sands that were in the column, and the iron oxide coated sands were actually the sands with iron oxide coatings that we coated ourselves. So in terms of size, they were the same size. Now in terms of surface area, I don't know if the iron filings had more surface area than the iron oxide coated sands, and we didn't do like a surface area analysis on those. This is all like sort of preliminary data. Most of it's unpublished. So.
Okay, way in the back there.
Hi, Ali. David was saying that his work at Discovery Bay was showing that sunlight photodegradation of chemicals was an important mechanism. Have you thought about looking at sunlight in these systems as well, given that the iron was effective in bacterial removal and would also potentially be a semiconductor? You might get some benefits from sunlight exposure to the iron.
I mean, I'm not sure how we would get sunlight into the subsurface. However, we have discussed, in particular with LA Department of Water and Power, some system that's a coupled wetland and then infiltration system. So there would be some treatment of the stormwater where you could take advantage of sunlight and photochemistry, and then after that occurs, you could infiltrate the stormwater to take advantage of sunlight and filtration.
Okay, well, through the miracle of 3 really efficient talks, we're right back on schedule. Let's thank our speakers.